Hey everybody, my name's Chris and welcome to the Stress-Free NP. This channel is dedicated to skills no matter what your title is. We're going to take you step by step and create a systemic approach to help you and your practice become stress-free. Always remember to like and subscribe so you don't miss any weekly video updates, and let's get started. Hey guys, so today we're going to learn about phlebotomy. Now, phlebotomy is the skill of drawing blood. This skill is very difficult and takes a lot of practice, but I'm going to break it down for you step by step so we can go through all the different details. Okay, so the first thing that we want to talk about when we're talking about phlebotomy is we want to talk about anatomy. So the anatomy meaning the anatomy of the arm. All right, so we have the forearm here, we have the antecubical area, we have the wrist, we have the hand, we have the fingers. All of these spots we can draw blood from. So we're going to start at the top and work our way down. At the top, you have 4 different veins, all right? So we have on the lateral aspect of the arm, which is the outside part, we have what's called the cephalic vein. Now, the cephalic vein is a larger vein and it's very easy to anchor. So this is a vein that you're going to use quite often when you're drawing blood. In the middle here, you're going to have 2 antecubical veins, and they're going to come down in a V formation just like that. There's going to be one here and one here. These veins are also very large. They're very easy to anchor, and they're going to be one of the most common veins that you use when you're drawing blood. The 4th vein that we're going to talk about is the basilic vein. And the basilic vein is on the medial aspect of the arm, meaning the inside part of the arm. And that vein is right here. This vein tends to be very small. It's not very easily anchored, and it moves and rolls a lot. So this is a vein that we're going to try to avoid if we can. If there is no other options, obviously, then we'll be able to stick this vein. But these 3 veins here, the antecubical and the cephalic, are going to be your first options when you're looking at veins. In your hand, when you apply a tourniquet, the tourniquet is going to go on the bicep when you're looking at the antecubical, the cephalic, and the basilic vein. When you're looking at the hand, in order to get the best blood flow into the hand, you're going to want to put the tourniquet on the forearm. And the hand veins are going to come down like this into the fingers, and they're going to pop up. Some veins that are better to stick than others are the veins running along the medial aspect of the wrist, and the 3 veins that wind up right here. Okay, the hand does tend to hurt a little more than the upper arm. The veins tend to roll a lot, they tend to be smaller, and they tend to be a little squiggly. So we're going to try to avoid the hand at all costs, but as we know, sometimes we don't have an option. So now that we went over the anatomy, let's go over all the equipment that you're going to use. The first thing that we're going to go over is the needles. This is a standard butterfly needle. They come in 3 different gauges. This is a light blue, so this is a 23 gauge needle. This is the most common needle that you're going to use when you're doing phlebotomy or blood draws. The other gauges are 21 gauge, which is green, and 25 gauge, which is dark blue. 25 gauge is smaller. So what does gauge mean? Gauge is the size of the lumen of the actual needle itself. The higher the number, so 25 gauge is smaller, the lower the number, the 21 gauge, is larger. This is different from when you were talking about French. French is the lumen of a catheter or an NG tube, and that's the opposite. The larger the number of a French is actually bigger, and the lower number of a French is actually smaller. So that's the difference between a French and a gauge. But when we're talking about needles, it's always measured in gauges. So now we're going to go over the different equipment that we need at the bedside or in a doctor's office or a lab or wherever you are in order to make a proper blood draw. Sometimes this equipment comes sporadically in the lab and you have to collect it. Sometimes you can grab a kit like this, which is called an IV start kit, but all the materials are the same. So if we open this up, what we have is a set of gloves, a tegaderm, alcohol pads, a tourniquet, gauze, and a chlorhexidine squab. When you're doing blood draws, you can either use alcohol or chlorhexidine. The difference between the two is when you're using alcohol, you have to rub in concentric circles starting from inside to outside. When you're using chlorhexidine, you don't have to use circles. You can just scrub wherever you're going to draw. So that's the difference between alcohol and chlorhexidine. The next part is the tape. There's 3 different types of tape. There's medical tape, which is your standard tape that we see all around. There's paper tape. Paper tape is used for people who are older, who maybe don't have the best skin. Their skin might rip easily. This tape is used a lot better. It doesn't really pull hair as much, and it's a lot more comfortable tape to use. The next tape is silk tape. This is the toughest of the tape. This is the hardest to get off. If you're really trying to keep down a dressing, this is the tape that you're going to try and use. When you have your standard needle, there's going to be one end that's going to look 2 different ways. It's going to have a lure lock on it, and you're going to be able to attach your vacutainer to this spot. The vacutainer looks 2 different ways. There's this vacutainer with no needle inside. So the other part that this can look like can have a needle attached to it with a sheath that's made of rubber. And what that goes in, and you screw in the lure lock, and now you have the needle inside the vacutainer. The second one is the one that we're going to use for this particular butterfly. And this vacutainer has a blue top and it already has the needle inside of it. So what we're going to do, we're going to screw on the vacutainer. Now, this little rubber part is a needle. So we don't want to stick our fingers in here and touch it because it will push back and there is an actual needle in there. The next thing we're going to talk about, and probably one of the more important things, is the tubes that we use. The tubes that we use are what goes in the vacutainer itself in order to collect the blood to be sent off to the lab. And there's many different tubes, whether you work in a lab, hospital, doctor's office, there's a lot of different color tubes that you're going to use, and we're going to go through the most common. So the first thing you have to realize when drawing blood is that there's an order of draw. And this is the order that you're going to draw the different color tubes in when you're drawing blood. And the reason we have an order of draw is because each tube has a different additive. And you don't want to mix additives into the different tubes, and I'll explain why in a little bit. Now, we have all our different tubes here. We have light blue, we have red, we have yellow, light green, dark green, lavender or purple, and gray. We also have a set of blood cultures here. Blood cultures are really only going to be used in the hospital, and these are large collection vials that we use to collect blood in order to grow bacteria. Now, when you're drawing blood, the first thing that we're going to do is we're going to collect our tubes. Now, we're going to collect our tubes in what's called a rainbow. If you're ever in a hospital and they tell you draw a rainbow, this is what they're talking about. They want you to draw all the different colors in order to get a variety of different tests, and we're going to go over the different tests for the different tubes. The first tube we're going to talk about is the light blue tube. This is the first tube you're going to draw in your order of draw. Okay, the thing that we have to realize with this light blue tube is that there's a line right here. And this line is to indicate how much you have to fill the tube. The lab will not accept this tube if it's not filled all the way up to this line. And this blue top tube is used for things such as PTINR and APTT. Basically, all your clotting and coagulation factors are drawn from this tube. The second tube is your red top tube. This is really more for your specialized blood draws. Sometimes we use it as a waste when we're wasting blood, but most often, if you're calling for a red top tube, it's really for a specialty blood draw, not really a common tube that we use. The third is the yellow top, or what's called a serum selective tube, or SST. Now, this tube is one, it does not have an additive in it. What it does have is this gel at the bottom. When you put blood in this tube and send it off to the lab, they're going to put it in a centrifuge that's going to spin it at very high rates. And what's going to happen is this gel is going to transfer all the way up to about halfway up the tube, and it's going to separate the cells, the white blood cells, red blood cells, platelets, all the different cells of the blood, and the serum. And the serum has all the electrolytes in it. So this tube is where you're going to get all your electrolytes from. Okay? Now, this tube comes in a variety of different colors. There's the yellow top. There's also the speckle top or the tiger top. These are all SST tubes, and they're all used to run chemistries. The next tube we're going to talk about is the mint green. The mint green tube is like the red. It's another specialty tube. Now, it does have the gel at the bottom, and it is used to draw chemistries. So different chemistries that can be used, such as troponins or ammonia, depending on what protocol your hospital follows in their lab, that's what you're going to use this tube for. If you are drawing an ammonia level, it has to be placed on ice. When you send it to the lab and you put it in your biohazard bag, you need to place this tube on ice when you're drawing an ammonia level. The next tube is the dark green. This is another type of chemistry tube, and this tube is used to draw chemistries in other certain hospitals. It's the same tube as the SST tube. Like I said, it just depends on what your hospital uses. We come to our lavender tube, and this is one of the main reasons for our order of draw. The lavender tube has what's called EDTA in it. And EDTA is an anticoagulant. So unlike your green top or your SST tube, the blood in these tubes are going to clot. The blood in this lavender tube is prevented from clotting using the anticoagulation of EDTA. Now, the reason for the order of draw is when you put this in the vacutainer, some of the additive might get inside the needle and the tubing. We don't want to put an anticoagulant in these types of tubes if we draw the purple top first. That's why it's all the way at the end of the order of draw. The purple top is used as a CBC, which is a complete blood cell count. Now, the complete blood cell count does stuff like your white blood cells, your red blood cells, your hemoglobin, your hematocrit, your platelets, and all that stuff. And that's the most common test that you're going to draw with this purple top tube, and that's a CBC. The last tube of your order of draw is a gray top. The gray top is used to draw a lactic acid level. This is really more, again, in the hospital setting when you're drawing lactic acid levels in settings such as sepsis or any type of acid-base imbalance where you need to know, is this a lactic acid acidosis or is it something else that's metabolic? Also important to know, when you are drawing blood cultures, okay? And this is in the hospital setting. Sometimes you're going to draw blood cultures in the outpatient setting for follow-up, but mainly in the hospital setting. Blood cultures are used, and they're used during sterile technique. Sterile technique means once you clean the arm, you're not allowed to touch the vein again. You have to stick with the needle. All right. Now, when you have your tubing and your butterfly needle like this, there's air in this tubing. So when the first tube that you hook up to this vacutainer is gonna get a little air in it. The two types of blood culture tubes that we have, the blue top, which is aerobic, meaning that it grows bacteria that breathe oxygen, and an orange top or an anaerobic tube. That means the bacteria that grow in this tube do not breathe oxygen. So when you're drawing blood cultures, and this is the first tube that you're drawing, you want to draw your aerobic tube first. So when the air comes from the tubing into the culture, you're not contaminating the anaerobic tube. Thank you guys so much for watching. Really appreciate it. If you liked the video, please like and subscribe so you don't miss any weekly video updates, and we'll see you next time.